Thank you so much for that really warm introduction. And thank you to Siri and all of the folks at CFRI for giving me the opportunity to talk today. So I'm going to try and do this quickly, but give you some pearls to take home. This is work in progress, genetic therapies. I don't have any answers or solutions. We have approaches, we have a lot of opportunities, and we're going to learn a lot in the next year. These are my few disclosures. None of them are conflicts with today's talk. So what I'd like to do is talk a little bit about the history of genetic, or what I'm going to call nucleic acid-based therapies for CF. We'll talk a little bit about the basic biology and approaches to the delivery of nucleic acids, because that's been a major stumbling block, I think, for the field if we look back historically over the last couple of decades. Talk a little bit about the challenges to success. The status of clinical trials will be very brief, but we'll give you an overview of what's happening right now and what sort of tools and programs have been developed so that we can complete these studies, and in particular, how we can engage, as Megan talked about previously, the group of people who are not eligible for modulators and really, really need genetic therapies. So we can go back over 30 years now to the cloning of the CF gene as the beginning of genetic therapy, because back when I was a resident in my training, this came out. The gene mutation was identified in 1989 by 3 labs who collaborated beautifully. You notice Francis Collins in the middle of this picture. And this was published in Science with the identification of the F508del mutation. And one of the children who was followed in Pittsburgh at the time wrote this after hearing her mother talk about this. "Today is the most best day ever in my life. They found a gene for cystic fibrosis." Cute and heartwarming, but it gives you a sense of the kind of hope that the community felt when the CF gene was cloned. And it turns out there's been a lot of progress based on just this seemingly simple identification of the gene. So let's define genetic therapies for those of you who are not familiar. Genetic therapies are approaches that can do one of a number of things. One is to fix the DNA. That's gene editing. That's the holy grail. That's where we probably have the longest to go to get there. Currently, you can try and replace the DNA via gene therapy or gene transfer using an intact, normal copy of the CF gene. Or you can use RNA, as it's called RNA therapy, where it's just the messenger RNA that encodes for the CF protein and replaces disease cells with a normal copy. So those are really the 3 fundamental approaches that are being pursued, and they're at varying levels of development. Gene editing is very much in the lab. And some of the talks today by Dr. Jonas, Dr. Amaya, Dr. Porteous, and then room next door addresses some of the approaches that are being used there. The field right now for clinical trials is at the DNA and RNA level. So we know that this is a highly potentially transformative approach. It's exciting, it's a new therapy, but we have to be sanguine about this because there's been a long history here. Rick Moss and others were involved in this adeno-associated trial in the upper left-hand corner. It was a trial back in the late 90s and early 2000s that looked very promising, but turned out not to meet its endpoint, was unable to show clinical benefit. Since then, there have been tremendous scientific advances, and this just lists some of the things that have been done, approaches or diseases where we have use of either mRNA or DNA therapies, or in some cases, gene editing to actually transform diseases. The gene therapy for inherited blindness, a gene therapy for spinal muscular atrophy, a gene therapy for sickle cell disease, and then really remarkably in May of this year, a publication on in vivo gene editing for a rare metabolic syndrome that caused hyperammonemia. They did it in 7 months. It was remarkable and a real tour de force and testimony to the power of science. So Siri alluded to scientific support. We need scientific support because this is not an easy task. It's going to take time, and we need funding to get it done. So the historical perspective, really, I'm going to start more recently. In the right-hand side, you see we had human studies with lipid DNA complexes back in the 2013 to 2016 era, a several-year hiatus. And then most recently, in 2022, 2023, studies with adeno-associated viruses, lipid mRNA complexes, and recombinant herpes viruses. The timeline in between there includes a number of attempts to cure CF with genetic therapies, included studies with early lipid DNA complexes done in England and studies with adenovirus back in the early to mid 1990s, and then one type of adeno-associated virus that I showed you the publication earlier. What's happened since then? What's been in the intervening that's allowed this to move forward? Well, A, is we've learned a lot about the basic biology, and B, the technology has really moved along when you think about lipid complexes and how they can be developed. So CRISPR was discovered in 2014. You had studies with gene editing in the lab in 2015. You had a silencing RNA drug approved in 2018. Then the vaccines, the transformative vaccines with the COVID epidemic by Moderna and Pfizer. And then we move into the current phase of studies in CF on the far right-hand side of this panel. And so where are we? We have all these treatments for CF. This shows the schematic, the pathophysiology of CF. We have all these approaches. Where we really want gene replacement, gene editing, so that all people with CF have a therapeutic option. And the drug development pipeline and the CF Foundation is rich with programs that are attempting gene therapy or gene editing. So all those in the dark blue in the lower left, some of the clinical ones I'll mention, but there's a whole slew of them in the middle that are in the middle here. These are in early stage, basically preclinical studies, trying to develop an efficient construct to deliver an mRNA or DNA or gene editing to the cells that we need to correct to have an impact in CF. So what has to happen? What's the pathway? And I think many of you may be familiar with this, but when we think about it sort of from the 30,000-foot level, we have to start with the gene. That's all genetic therapies start that way. We have to understand something about its expression and its function. Then we have to come up with a way to deliver it. And that, I think, if we're honest, has been the biggest challenge with the field. We still don't have an optimal way to deliver the normal CF gene into the appropriate cells. And once we have potential candidates, we have to demonstrate safety and efficacy. We'll talk about that in a few minutes. We have to design human studies that take advantage of a limited patient population and are really focused on safety and early efficacy. We have to execute those studies. And then ideally, in the long run, we have regulatory approval from the FDA or the EMA in Europe. So what are some of the advances? There are certainly advances in laboratory models. You've seen pieces of those today with things like CF ferrets and CF pigs. So laboratory models that can be useful for demonstrating safety and efficacy. We have approaches to innovative trial designs. I will talk a little bit about that in a few minutes. And then we need the infrastructure and tools to engage people with CF for study participation. And we've made some nice progress there with support of the CF Foundation. I'm not going to spend a lot of time about genetics, but suffice it to say we have these different classes of mutations, and the group that's really in highest need are the individuals who have class 1 mutations with that do not respond to modulators. Many of the mutations in these classes are modulator eligible. We still have 10% or so of the population that doesn't have an effective modulator drug and really needs one. So the genetic medicines are so attractive because they have potential to provide therapies for all people. Modulators will improve for the population of people where the drugs work by repairing the CFTR protein. But genetic medicines could restore, fix, or replace the gene, and that would be relevant to all people with CF. I mentioned that we've learned some things about the challenges to delivery. I think this is really important that we understand the epithelial biology that goes into the challenges of getting into the cell. We know that CF mucus is sticky, thick, and acidic. We know that there are immune cells that can react against whatever our gene transfer material is and neutralize the therapy. Unlike some cells, some parts of the body, the lung cells turn over. They turn over probably every 120 days. And so unless you target the stem cells that are at the root of the progenitor cells for the epithelium, we're going to need to deliver these compounds on a repeated fashion. And then the CF gene, as was alluded to earlier, is a big gene. So it's not necessarily so easy to do. We won't go into great detail about this. There was a nice talk earlier about ionocytes and how important they are in regulation of the airway surface liquid. But this schematic shows a cross-section of the airway. And what you see are the various cell types here that are involved in regulation of the airway surface and allowing mucus to get cleared out of the lung. So it includes club cells, goblet cells, ciliate cells, and then these ionocytes, which are a rare part of the population, and then stem cells. So in an ideal world, and as this field evolves, we hope that we'll have something that targets these basal stem cells, because if you corrected the CF gene in a stem cell, it will self-renew, and the cells will be corrected for perpetuity, and you'll end up with essentially a cure. So when we think about genetic therapies, it's important to think about the distinction between them and modulators. This nice slide that Kara Fauché had created shows the difference in the size of trikafta of messenger RNA, and the analogy is a kumquat to a watermelon. The size difference is really quite substantial here, 1430 grams per mole over 2 million grams per mole. So very big constructs that have to get into the cell. A small molecule like trikafta can diffuse into the cell from the bloodstream. It goes out into the circulation. Very different approach when you're talking about genetic material, which is much larger, and the cell has its intrinsic barriers to prohibit that material from entering into cells. So if this is the approach to either give RNA or DNA or gene editing, how are we going to do this? I think one analogy I like to use is vectors. There's a lot of vectors in the world. We have vectors like mosquitoes. They're a vector of bad things. We have ticks. We have a lot of ticks in Pennsylvania. I'm not sure about in California. A lot of deer ticks and a lot of Lyme disease, thanks to deer ticks. So what do we do with DNA or RNA? How do we get them into cells? And thankfully, there have been a number of nucleic acid vectors that have been developed. And I just list some of them. We're not going to go into great detail. They all have their pros and their cons. But suffice to say that all of these have demonstrated, at least in the lab, that you can put either a normal copy of messenger RNA or DNA into airway cells. And this preliminary data is what's led to clinical trials. So let's say you have a vector. What do we do next? Well, obviously, you want to make sure it works. So there's a variety of ways to look for in vitro expression using immortalized cell lines and, more importantly, polarized bronchial epithelial cells. And then if there's success there, we go into animals. And expression in the airway is a really high, important milestone to meet. And then we have to show safety in animal species before you can project and go into people with CF. And CF ferrets and CF pigs and other large animal models can be very helpful here. There are a number of issues that make this more complicated than just thinking about any airway cell. We have target cells like the basal cell versus the ciliated cells. We have a pretty good idea of the percentage of cells, the efficiency that we need to have a physiologic benefit. It's probably in the order of 10 to 12%. We have to think about issues of integration and whether that integration is in the right place, in the right cell, or is it random and could cause something like malignancy. And then think about shedding. So the safety issues associated with shedding of a piece of a virus or a whole virus. Thankfully, these preclinical animal models, we've learned a lot as we've gone. We have a good sense of what animal models are a good bar for doing these studies and moving them to humans. So we want to mimic the delivery. They're done by aerosol for the most part. And this slide just gives you an idea of expression using a fluorescent label. This is the airway here. These are the cells that line the airway. And you see in this experiment, very robust expression of a fluorescently labeled protein in the cells. So that's an important way to do it. One of the things that I try and address particularly with patient groups and patients that I counsel in clinic is about what the clinical trial process looks like. So what data do we have before we go into humans? Well, first, we really have to have a delivery vehicle that might be effective. We're not going to study something that looks like it's not going to work. So setting a reasonably high bar for the preclinical studies is important. And then we have to be very thoughtful about considering the risk of these therapies to human. We have to consider the risk of the genetic material and the risk of the components of the delivery, that is the vehicle, either the lipid nanoparticles or the virus in the current generation. And then we have to think about risk of the procedure. So some of these procedures, some of the studies require bronchoscopy or need bronchoscopy to learn a lot. So there are some risks associated with those procedures. We have to be thoughtful and careful about how we do these. But I want to make it really clear that as an investigator and in our investigative community in CF, safety is always the highest priority. So we don't take risks in people without having assurance, the best assurance we can from laboratory and animal studies that there is some benefit. The second important thing to remember is that there are multiple layers of oversight to the clinical trials that go on in CF and other diseases. First of all, the study sponsor has obligations. They don't want to do something that's too high risk. They have that in their mind. The FDA reviews all studies before they go into people. In the CF community, we have a Therapeutics Development Network that does scientific review of protocols before they go into people. Once the trials start, the Data Safety Monitoring Board is doing real-time analyses, real-time looks at the data to carefully make sure that the studies are proceeding with adequate patient safety. The institutional review boards have a say in whether a study is reasonable to proceed or not, and whether it should continue when there are any amendments. And then we have the site study teams and the participants. And at the individual site level, they're both really important for communicating issues or problems and sharing those in real time so that the rest of the community is aware and can make amendments or changes to protocols as they go forward. The FDA has been, until recent changes and hopefully in the future, been very supportive of developing therapies for rare diseases like CF. They look at things on a risk-benefit profile in the patient population, and they really ask for demonstration of how patients feel, function, or survive. That's the bar. The company needs to show benefit in how people feel, function, or survive. And the amount of regulatory flexibility that they give is proportional to the disease severity, the rate of progression, and the degree of unmet need. So if you take a disease like infantile onset spinal muscular atrophy, which is fatal early in life, that is a very severe disease, and there's lots of regulatory flexibility and probably a higher level of risk-taking for an early childhood fatal disease. If we think about CF here in the middle, we really have two populations. We have the modulator ineligible population, which we know is not doing as well, has a higher degree of unmet need, and then the CF modulator eligible population, which doesn't have severe disease at this point. And maybe in that population, we need to be more conservative in how we do clinical trials. So the FDA is thoughtful about these things, and hopefully as their administration turns over, we'll continue to have that. One of the beautiful things about the FDA and to some degree the EMA is there's an ability for disease communities to communicate with the regulators and give input on things like the patient population, and in CF, how the patient population has changed, where the areas of unmet need are. And then the CF Foundation, other private disease foundations can advocate on study design and outcome measures. And the CF Foundation and the Therapeutics Development Network has done that now for several decades. It's been very helpful for the development of modulators, and I think will be helpful as we move through the development of genetic therapies for CF. So I mentioned that there are studies that have been done that didn't meet the primary endpoint, but I think it's important to know that these studies were done very safely. And they give me great hope that we can do studies in dozens, involve dozens of people with CF and get them done, have the right safety mechanisms, and not have any bad outcomes. So this is the results of study that Rick Moss, who's here with us today, published in 2007. It shows the lung function changes over time, comparing in the AAV-treated group to the placebo group. And you see they didn't meet significant difference over time. And so this was abandoned, not taken on to phase 3. But the safety parts of this were a great example of how we can do studies of novel therapies in a patient population like CF. And this was in the pre-modulator era. I think if we reflect, there are some early lessons on safety and efficacy. The studies with adenovirus and adeno-associated virus, and at least one of the mRNA platforms, the MRT5005 from Translate Bio, we know that they weren't efficient enough to improve FEV1. So there was no physiologic benefit to these, unfortunately. So we learned that. And as I mentioned, there's been a lot of work to try and refine the vectors and make them better and more efficient. And the current studies will hopefully find that one or more of those agents meet that bar. We know there have been symptoms associated with inhalation That's always been the case with CF therapeutics: cough, airway irritation, bronchospasm, acute decline in FEV1. That's happened in the past in some people with hypertonic saline, for example, and it happens with some of these gene transfer vectors. We know from the messenger RNA vaccines that systemic symptoms like fever, myalgias, headache are very common. Most of the people in the room have probably endured those symptoms with the vaccine. And not surprisingly, these things can happen. The same symptoms can happen with some of these genetic therapy vectors. So as a result of that, we've learned that we may require some modulation of the immune response for these agents to be tolerable. So in order to have repeated dosing, you may need steroids or other immunosuppression to blunt the immune system, but yet let the vector do what it needs to do to help correct the airway cells. So where are we with the current genetic therapy trials? These are all in various stage of progress. The top mRNA studies, the Vertex 522, Arcturus has a compound called Lunar CF, and Recode RCT2100. These are all through phase 1, early safety on single and multiple dose, and are now in phase 2. I won't talk about antisense oligonucleotides today, but Splicense has a protocol that they're trying to finish. And then there are a number of so-called gene addition therapies using recombinant viruses. The 4D program is the most advanced. It's 4D710. That's also at phase 1/2 and making progress. Spirevent is another recombinant adeno-associated virus. Crystal Bio has a herpes simplex virus that was approved for use for a topical dermatologic condition. They're now testing that as an airway. That's in phase 1. And then in Europe, Boehringer Ingelheim has a recombinant lentivirus. This is thought to be integrating and may provide a more durable effect. That trial is not up in the United States at this point. So I'm just going to give you one example without any bias that this is, and that any of these are better than another, but it gives you a flavor for what sort of data has gone in on the preclinical side that led to the human studies. So the Recode compound is RCT2100. What this study shows, using a tried and true gold standard in vitro model of CF epithelia, that as you increase the concentration of RCT2100, you get more chloride current, shown here on the y-axis, and it approaches, this is the 20% of wild type. This is with ETI, so it provides some good evidence for restoration of chloride transport in airway cells. And then they took this into ferrets and tried to show that you could improve mucosoid clearance. I'll skip the middle of this, but suffice it to say that when you dosed with RCT2100, you have an increase in clearance compared to here, the baseline. This is delivering a radio-labeled compound into the airway and watching it move out of the ferret airway. It moves slowly here, moves much more robustly here. So again, animal model data to suggest that these work. Gene editing, I mentioned, is back in its infancy. It's still very much in vitro and in animal models. The attempt here is to edit the gene by targeting it, cutting it, permanently repairing the CF gene. So the goal here is to get basal stem cells. And it's very clear from work in the respiratory community and in other communities, liver, bone marrow, other diseases, that the mechanisms are there. The basic biology, the understanding of how you edit genes is very clear with having a RNA with a guide and a plasmid construct in some cases, and then a way to develop the editors, deliver the editors as a group of compounds, a group of molecules that would then take a gene and edit it to make it normal. There's some proof of concept for this and some nice papers that were published in the last year or two. This is an example from Prime Editing. They looked to target the F508 del mutation airway cells. They delivered a construct of varying editing machinery and showed that you could restore chloride secretion in vitro, shown here and shown here. So they're good example of how this can be done in vitro. This proof of principle is going to be carried now into animal models. And ideally, if it works and is shown to be well tolerated by animals, it would then go into humans sometime in the near future. So I'm going to just pivot a little bit to clinical trial design and how they're done and what the community's doing to try and make it easier to get drugs through the pipeline of clinical research to be approved. We know that FEV1 is the gold standard outcome measure for therapies in CF. And this slide shows what happens with the modulators, ETI and ivacaftor. Vanzacaftor is very comparable to ETI. We know that there were first generation, if you will, modulators that were not so effective from a FEV1 standpoint, but still had some degree of clinical benefit. We hope that nucleic acid based therapies will have at least as good as here, but if we would be really happy if we reach this point. If we have 5 to 7% increase in FEV1 in a population, I think that would be considered a win for us and would provide early therapy that then could be evolved the way modulator drugs were evolved. We had modulators that were effective but not highly effective, and now we have highly effective modulators. So the clinical trial requirements of these different classes, the modulators and the genetic therapy is very different. The top here shows the progression from phase 1 to 2 to 3, safety and efficacy, safety primarily here with a little look at efficacy. Phase 2, efficacy is a higher priority, and then phase 3, really efficacy as in a larger population. If we compare what the needs are for small molecules, like the modulators and what we've been doing thus far with the nucleic acid-based therapies, the number of patients required to demonstrate benefit in a lot of the early phase studies was much higher than it is anticipated to be in people with the nucleic acid-based therapies. This 3+3 design starts with a low dose, demonstrates safety, moves to higher dose, demonstrates safety, move to higher dose. So it's very much dose finding with small numbers of individuals. And assuming there's some success there, we'll move into expanded cohorts of 6 to 9 to begin to look for a group of efficacy signal. We hope that phase 3 trials will actually not require more than 80 or so participants, in order to learn whether the therapy works. How are we going to effectively do that? The main approach, unfortunately, is going to require targeting of a relatively small patient population. Megan talked about this earlier. We have a large number of adults who are eligible for modulator, and then about 1,600 adults with CF who are not eligible for modulator. If we look at those and include those who are intolerant or have limited benefit, and we think about lung function criteria, FEV1 40 to 90, and not having had a lung transplant, we're still talking about probably 800 to 1,000 people in the United States to do these clinical trials. It's not many when you compare to the pool who are eligible for modulator studies. So Nicole Hamblett and JP Clancy and others have really worked on thinking about how we can be creative about trial design. The standard clinical trial enrolls people and puts them into an active treatment and a placebo and compares the outcomes. That's been the gold standard for years. When you have a rare disease with a small patient population, the FDA has been supportive of doing trial designs that allow for an external control. So this is a group of people with the disease who are very well characterized, who you can use as your part of your placebo group. So it would potentially allow you to randomize instead of 1-to-1, randomize 3-to-1 or 4-to-1. You still need some randomization, but it's helpful to be able to have fewer subjects by having an already created external control group. And the CF Foundation is in the process with a nice study designed by Deepika Polanini and Nicole Hamblett to basically create that external control population. So the goal here is to collect contemporary data in a prospective study from people with CF who are not eligible for modulators or are not taking modulators, to get a careful look of what their symptoms, their pulmonary function, their disease trajectory, their exacerbation frequency, all the clinical things that we follow, characterize that group and create an external control population that could be used by a variety of different drug development sponsors in the genetic therapy space to use as part of their placebo group. So it would allow us to get studies done more efficiently and get to our goal of a cure with genetic therapy sooner than we would otherwise. The other approach has been to engage the CF Therapeutics Network. I think most people in this room are familiar with it, but the CF Foundation created a therapeutics network over 25 years ago now. Started with a small number of sites and expanded to where now we have 80 sites across the United States. And those different groups are focusing on all aspects of genetic therapy for CF, both genetic therapies, symptomatic therapies, and then really working hard to think about how we allow people with CF in all parts of the country, rural, urban, big center, small center, to get into clinical trials, particularly those of underserved racial ethnic groups. So I'll go through this quickly for the sake of time, but there's a lot of materials that have been done and developed over the years, working with sponsors in the FDA, things like regulator workshops, compound review prioritization, materials for the care centers to educate the care centers, because not every care center does clinical research. So we want care centers that are not research centers to engage and help participate to recruit and enroll patients for us. So to facilitate this interaction, this collaboration between research centers and care centers, we created a regional cooperative initiative. This was to build research knowledge in the CF centers, improve the access to clinical trials, and increase referral opportunities. So the goal is that every CF center is plugged into one of these regions. Some of them are big in the West where people are spread out. Some of them are really close together in the Northeast where there are lots of people. But the goal is that the care center would know who to refer patients to, that have regular phone calls, regular education sessions, so that the care center people can identify people with CF who are eligible for clinical trials and get them referred to a research center so that they could participate. We want equity, we want diversity, we want inclusion, and this is, I think, a very solid attempt. The number of referrals from care centers to research centers is gradually going up, and we hope that that will continue to allow us to get these studies done quickly. So for those of you, people who have CF, how can you participate? What can you do to help this mission that was so critical for us? You can certainly discuss research with your care center providers. Ask them. Don't wait for them to bring it up. Ask them about research. What's going on? What are the opportunities for us? And then the CF Foundation created a clinical trial navigator. It's a single person, single email that people with CF can email and set up a time to have a phone call and say, this is who I am. This is what I'm about. The navigator asks questions to determine eligibility for studies, and then can help identify care center or research centers nearby who might be able to help facilitate them getting into a clinical trial. They have a web-based approach to doing it on the CFF website. There's a form that can be filled out as well for the same sort of access to get engaged in the community. So we know that developing new therapies for CF in the genetic space is going to require everybody. It's going to require the care center teams. They absolutely are going to have to be the frontline people who help identify and make referrals. We don't think we have the perfect answers yet. We don't know that. So until we do, we need basic and translational investigators to work at the fundamental level so that we have options. If the worst case scenario, none of these current trials turn into a therapeutic, we want to have options. And we need basic and translational investigators to help do that. Our research coordinators are critical. At our center, they are the front line for clinical research. The people with CF in our center know them. They know their name and who they are. They establish relationships. It's invaluable to help recruit and get people engaged in clinical research. And then we need people with CF. We can't have people sitting on the sideline. If we're going to develop a cure, particularly a genetic cure, it's going to take a lot of effort and a lot of people. It may only be 100 people, but if there's only 1,000, that's 10%. Very rarely is 10% of a population enrolled in a clinical trial. It doesn't happen that way in a lot of other disease cases when you think about it. So that's where we are. And what I've tried to do, I hope I've done in the last 45 minutes, is to talk about how we identified a gene, a group identified a gene, and that enabled the development of modulators, but also the design of nucleic acid-based therapies. We've learned we have challenges to the delivery of those nucleic acids, and increasing understanding of that and developing new delivery methods has now progressed to clinical trials that in the next 6 to 12 months, we should have some readouts and a sense of whether any of those have potential to move into the clinic. Clinical trials are done with incredible safety and scrutiny. There's a lot of oversight, and safety is really the highest priority. I think we should get past the days of people being afraid that participating in clinical research is a risky endeavor. Does it have risks? Yes. Those risks are very calculated and very well communicated to people before they begin clinical trials. And then I think I'm really optimistic we have the right infrastructure. We have the partnerships with industry that's continuing to work really hard to help develop new trials. We have the infrastructure on the care center team side and on the clinical research team side. And this continued engagement, education will hopefully get us to where we want to be. I've been working in CF for a long time. I won't say how old I am, but it's been 3 decades. And the modulator population has transformed our clinic, but we still have a group with very high need. And they struggle every day like we had before modulators. We really want something for them. And so we want everybody to jump on board and do what we can. So I'm just going to end with a quick acknowledgement slide. I've had a pleasure working with a large number of people in this genetic therapy space and at the clinical research space of the CF Foundation. And I mentioned a few of them. Dan Sigwart is a basic scientist who taught me a lot. And then Steve Rowe, JP Clancy, people at the foundation have been really helpful for helping us to educate our committees and help us move things forward. So I'll close there and thank you for the opportunity.